So I was going to do a couple of things relevant to the homework to get started. I did extend the due date mentioned on the discussion board at some point a few days ago that we'll make this due Sunday instead of Friday. And that's fine generally going forward. I'm happy to make them due on Sunday. In number 3, you do an alignment of 2 amino acid sequences, and then you did a significance test, like from the seqanalysis.r script that I gave you, to assess quality of the actual alignment. When you do that, remember that we take the 2 sequences we want to align, and we take the shorter one, the one that's no longer than the other, and we create random versions of that sequence. The code is in the seqanalysis.r file, but the idea is just to create a bunch of versions that are qualitatively similar to the one we are working with. Same rough proportions of the different letters, same length, that kind of thing. Do that many times, thousands of times, and for each time align to the original second sequence, which gives you an alignment score. Keep track of all of those scores. Store all of those scores. It actually comes out of the function that I provided. So suppose we have 5,000 scores. Each of them is for a false alignment. It is an alignment where the first sequence has nothing at all to do with the second sequence because we completely randomly picked the letters to put in it. So in hypothesis testing language, it's like we're forcing the null hypothesis to be true. The null hypothesis being that the 2 sequences don't align together. They have nothing in common. And our question is, if that were true, if the null hypothesis were true, how likely would it be to see an alignment score like we saw or something even stronger just by chance? And if that answer is small, if that p-value is small, then we probably have an alignment we can trust. So in this part, 3CI, you are to take all of those alignment scores for the randomized versions of your sequence and make a density plot, and then shade in part of the density plot. So I was going to show you how to do that. Let's come over here and open up a new file. So let's make up some data and make a density plot and shade in part of the curve. Let's create some just normally distributed data. So give me random normal observations. Give me 5,000 of them, say. And we'll say mean 0, standard deviation 1. Here's a picture, a histogram. Looks like that. And a density plot is basically take a histogram and put a smooth curve on top of it. A density in statistics, usually we are talking about a probability distribution. So it's for a continuous random variable. A smooth curve and areas underneath the curve correspond to probabilities. So suppose we have data like this. I could make a histogram. I want now a smooth curve on top of it. How do you get the smooth curve? There are different methods for getting a smooth curve. One is what's called a kernel density estimator. R has a built-in function called density, and here's what happens if I just call density x. It prints out information. So density, let's do question mark density, fits a smooth curve to a distribution. I'm going to go here to the base one. Computes kernel density estimates. You don't have to know what a kernel density estimate is, but it's a smooth curve. If I look down, so help files, we've seen these several times now. There's always an argument section. There's always a details section. And there's always a value section. The value section is what happens when you call this function. Is anything returned to you, or is just a plot made, or whatever? When you call density, an object is returned to you. So we don't really need, well, okay, we're going to get the object. So let me just store the object. Let me do it like this. dd is density. And before we go forward, let me make this histogram a little bit differently. The histogram is as drawn here by default. If you just ask for a histogram without any arguments included, we'll give you what's called a frequency histogram. So that means that the height of each of these bars is the number of times that you saw a number in that bin. So these are counts. The y-axis is frequencies or counts. You can make what looks like the exact same picture, but just scaled differently. If you say probability equal to true, so question mark hist, this argument right here specifies, do you want to scale this so that areas under it are like probabilities? Scale it so that the total area underneath it equals 1. The heights won't be counts anymore. Looks like this. I'm going to say probability equal to true. So the picture qualitatively did not change. It looks exactly the same. The only thing that has changed is the vertical axis. It's now called a density histogram. And all that means is that instead of the height being the number of observations in this bin, the area of this rectangle is the proportion of observations in that bin. Shape is still exactly the same. It's just the scaling's different. The reason I did this is because we're going to make a density, we're going to compute a density curve, and then we're going to overlay it on this. We're going to draw a smooth curve on top of this. You don't have to do that in your homework, but this is just an illustration of what you can do with a density. So I have stored density, class dd, it's a density class, it's a special class. What objects or items are available to me? I asked for the names of dd. There's something called x, something called y, and then some other stuff that we don't need. $x, here's the first 10 of them, and $y, here's the first 10 of those. So what those are coordinates for how to draw the curve. If you went to x-axis minus 4.30067, y-axis that, and put a tiny little dot, and then go to this one and put a tiny little dot, and go and keep going and keep going, connect all those dots, you'll be looking at a smooth curve. Looks like this. I can add the curve this way. If you have an existing plot in R, like we do, and you want to annotate it or add something on top of it, like let's put a point over here, or let's put some text over here, or let's draw a line, or let's draw a curve. There are some functions that add to an existing plot. One of them is called lines. Lines adds lines. We want a curve, but if you have sufficient numbers of points and they follow a curve, if you connect the dots, it will look like a curve. So I think I can just say lines dd, but I'm going to do it this way. Lines takes 2 arguments at the least, the x coordinates and the y coordinates, at which you want to put points and connect the dots. So if I say lines, this, let's make it pretty. Blue, line width is 2, so it's a little bit fatter line than it would be by default. You don't have to do that. Looks like that. So if you want to plot a density, I didn't have to overlay it with the histogram. I could have just done, I think I can just do plot $dd$x, $dd$y, and specify, please connect the dots. If I say type equal to L, that means connect the dots. Question mark plot. Here is one of its arguments. And if you say L, you get lines. There's other ones that you can do. You can do stair steps and other different things. So if I were to just do this, every time you call plot, you're going to get a new image, a new panel with a picture on it. So this is going to create just a brand new picture. There is no histogram anymore because we're just going to draw the curve. So you could do that to plot your 5,000 alignment scores. And then the other part of the question was shade in the tail corresponding to scores as big or bigger than the one you saw, because that's our p-value. This is a density. Total area under that thing is 1. Areas under it over subregions correspond to probabilities. So suppose we saw an observation of 2 in this case. Suppose this was our density picture, and our score that we wanted to assess was equal to 2. How likely would it be, if this is the distribution of alignment scores under the null hypothesis of incorrect alignment, how likely would it be to see a score of 2 or even higher? I need to basically integrate under this tail. That's basically what we would need to do. So to shade it, how do we do that? There is a function called polygon, which it draws polygons. I don't want a polygon. I want shade under this image. But the idea is, here's our picture, and I want to shade in here to the right. Polygon essentially makes lots of little rectangles and colors them. So if you give it enough points, it will look smooth. It won't look like stair steps. To do this, I have to specify the locations of all these little bin spots that I want to make this polygon picture with. So I have to specify all these little places. And for each one of those, I have to specify the value of the height that I want. So I can do it this way. We are going to do polygon, and I provide as the first argument the locations of those tick marks on the horizontal axis. Where are we going to put little polygons? That's what's going to go here. And then in the second argument, I'm going to put the heights for each of the corresponding rectangles. That's going to go there, and let's color it red. So here's one way I can do this. I only want tick marks at 2 or even higher than 2. I could take all the tick marks from the actual density plot. They're in $dd$x. Those are the x-axis locations where there are points in the density. What if I just took those where they are at least 2? So that's a bunch of coordinates on the horizontal axis that corresponds to points at or above 2. And let's say that the height for each of those is the density height, again, wherever x is at least 2. And then let's tidy this up. What I need to have happen over here, if I kind of zoom in on that, I need to say, draw a point here, and then one here, and there, and there, and there, and there, all the way down, and then go back down to 0. So like draw that polygon. There's a polygon looking like this, and shade that whole thing in. So what I'm going to do to tidy things up in these arguments is I'm going to say I want the first point to be 2, and then use all these points from the density, and then so we're going to start 2 at the bottom of the vertical axis, and then we're going to jump up, and I want heights that correspond to the density. And then at the very end, probably could get away without doing this, but I'm going to go ahead and make sure that it goes down all the way to 0 at the very end. So start at 2, and then all the x values, and then the very last point, let's just repeat the maximum value of $x. So these are all the x points that are going to define this region that I want to shade in. Over here, I'm going to start at x equal to 2, and I want to start at 0 for y. So I'm going to put 0 here, and then these will go with all of the x values from the density. And finally, I want to go back down to 0. And hopefully that worked. Yes. So that's shaded in. The region from 2 to the right. It's not perfect because the smallest one of these is slightly bigger than 2. I could tidy that out, but that's okay. So that's how you can shade in the region of a picture. Polygon is handy for that. Questions about that? The other thing that I wanted to touch on with regard to the homework is in the last question. Somebody asked on the discussion board this morning about these data, the QC spike data. Do we need any special background on these data? They are proteomics data, and we really haven't defined what that means. My answer is you don't need to know proteomics or anything to do this problem. These are just data for our purposes. These are omics or bioinformatics data. So they're going to look like an M by N matrix of numbers. One row per feature, one column per individual. Features in mass spectrometry-based proteomics are peptides, pieces of the proteins. So I have one row per each peptide that was detected in this collection of samples, and I have one column for each actual sample that I obtained. That's all you need to know to actually do this problem. Just treat it as a bunch of numbers. Rows are your features, columns are your individuals. So the description of the experiment that gave rise to these data, let's just spend 90 seconds on that. This is kind of a calibration experiment that some collaborators of mine did, where they took a bundle of salmonella proteins, and they had another bundle of not salmonella proteins. In this other bundle, there was proteins from horse, rabbit, several different non-salmonella species. And they divvied everything up into 3 batches. In the first batch, they put in or spiked in the non-salmonella proteins, quality control proteins, QC proteins. They put those in there with the salmonella at a high concentration. In the next batch, it's at a medium concentration, and in the last batch, QC proteins are at a low concentration. So for each sample, I've got lots of salmonella, and then I've got some of these QC proteins. The interest is in how we're trying to characterize the reliability of these instruments. So suppose you have contaminants in your sample. These QC proteins represent contaminants, basically. I'm studying salmonella. What is the impact of having more or less of these contaminants was the rationale for doing this study. Okay, let's open the data. So under data, these are proteomics data, so they are under the proteomics folder. And we'll save this. It's a CSV file. All right, is it CSV? Yeah, it's CSV, common delimited text file, so I can just open it in Excel. All right, so it looks like this. First column is a unique identifier for each peptide that was detected in the sample. We had a bundle full of proteomic material. We ran it through a fancy instrument, and we get out for each unique peptide that we think we identified in these samples, what was the, give me a number that quantifies how abundant that peptide was in each sample. So for example, row 1, this is a peptide. It happens to have this sequence. That's its amino acid sequence. You don't need to know that, but that's its mass. I also know for each peptide, we don't need this right now either, but later we'll talk about this. Each peptide is known to come from a particular protein. Proteomics, we don't, at least mass spectrometry proteomics, we don't usually work with intact proteins. But if you tell me you're studying salmonella and you saw that sequence of amino acids, we can look up in the genome of salmonella and figure out, well, that sequence corresponds to protein X. So for each peptide, we also have a code for which protein it came from. And notice that there are multiple representatives of the same protein. That's not uncommon. That's what you would expect. You have proteins floating around. They're big macromolecules. You break them up with an enzyme into smaller pieces called peptides, and then you're going to see, this piece sometimes, and you're going to see that piece sometimes, and so on. For our purposes in the homework, we're just treating it as if we have all these different features. Don't worry about the proteins for our purposes. Worry about the peptides. This column is underscore QC, tells me whether each of these proteins was identified as having come from the QC proteins or from salmonella. Most of them are going to be salmonella proteins, which means this number is going to be 0. But there is a small amount of these QC proteins floating around. And then the remaining columns for 8, 12 columns are the actual numbers. These numbers, you can think of them, well, we'll talk about it more later, but these are our quantifications of how abundant each of the peptides are. The first 4 of these, G0S1 up through G0S4, those are the 4 replicates from the first batch or first comparison group. And I think this corresponds to high abundance. Yeah, this corresponds to high abundance for the QC proteins. The next 4 are for medium abundance of QC and then low abundance of QC. Notice the NAs. Those are missing values, and those happen because, for example, that peptide was identified as being present in this particular sample, but not, say, in that particular sample, because there's an NA there. So I saw this peptide in one sample, and I got a number for it. I got a number, got a number, got a number, but we did not see that peptide in this sample or that one, and so on. And there's lots of NAs. That's kind of a common feature of proteomics data, that sometimes a peptide is observed, identified, sometimes it's not. If you're familiar with next generation sequencing, you extract genomic material, and in theory, representatives of the entire genome are there, but there may be some reads that you find for one individual, but not for another individual. Just because of the random nature of selecting the genomic material, you may not have gotten any representatives of this one from an individual. There could have been identification errors or sequencing errors. There's different ways that these types of things can happen. So we have lots of NAs. Yes? We are going to, let's see. Yes, so you have 3 comparison groups. If you think of the batches as being comparison groups, we have one group of 4 replicates where QC is high concentration, then another 4 where it's medium, another 4 where it's low. So 3 comparison groups. We might come in here and say, is there a difference for this particular peptide in its abundance comparing high concentration to low concentration? I could do that with a t-test, say, a 2-sample t-test. I have 4 numbers in this group. I have 3 numbers in this group. Suppose I tried to do that for this peptide. This peptide only has 1 number in the first comparison group, and it doesn't have any numbers in the second comparison group. So you can't do a t-test. To do a t-test, you have to be able to estimate the mean in each group, sample mean. You only need 1 number in each group to do that. But you also have to estimate a variance for the denominator of the test statistic. And so what I'm requesting in the assignment is make sure there's at least 2 non-NA values in each of the comparison groups. If there's not at least 2, I can't estimate a variance. Yes. So you could go, what you would want to do is go row by row and tally up how many NAs in group 1, how many in group 2, how many in group 3. And if any of those is 3 or more, exclude that row. All right. So those are the QC data. I asked you in the question to do a t-test, 2-sample t-test, comparing the high concentration group to the low concentration group. So ignore the middle one for the purpose of part D. Okay. What I wanted to start on today as new material is we're going to transition to talking about working with a data set like those data, the QC data. I have an M by N matrix full of numbers, and I want to explore it. I want to do testing. I maybe want to do prediction or classification. Let's start talking about statistical issues and tasks that would be common. One task and one topic that is important when we go start doing tests for each of our thousands of features. Is this feature different between cancer and normal? Yes or no? Is this one different? Is that one? And so on. That's doing hypothesis tests, p-values. Let's go put a p-value on all thousands of our features. So I have this script that's on eCampus. It's called testing.r. And it's just a bunch of code that'll walk us through thinking about issues in doing multiple testing. So I mentioned this before, but when you do a hypothesis test, classically, the methodology is set up to work where you have a scientific question, you do an experiment or collect data somehow, and you do a test. Then your p-value, if it's less than 0.05, you can confidently reject the null hypothesis, and you can say, I just did an analysis where I can say the probability I would have made a type 1 error is 0.05. If I rejected if the p-value is less than 0.05, probability I made a type 1 error would have been 0.05. But suppose you do, let's say you do 3 tests. We have some treatment that we're interested in, like a new experimental drug that our laboratory has developed, and we think it cures Alzheimer's disease or something, but we're not sure about dosage. We're trying to convince the FDA to let us sell this drug. And in order to do that, we need to be able to say, we did an experiment, and we gave the drug to people with Alzheimer's disease and placebo to others, and there was a statistically significant difference between the two groups. So ideally what you would do is go get a sample of people with Alzheimer's, randomize them to treatment or control, do a test, get a p-value. But suppose instead that we did it this way, we did a low dose, medium dose, or high dose, like how much of the active compound is actually in the pill. And for the low dose, we got treatment and control and a p-value. For the medium dose, we got treatment and control, some new people, and got a p-value. And here we got a p-value. Suppose we take these 3 p-values and we take the smallest one. And we say the drug works if the smallest p-value is less than 0.05. The probability of making a type 1 error, if you're going to go do that and reject if the smallest of them is less than 0.05, is not 0.05. The error rate, the probability of having made a type 1 error, is bigger than 0.05. If there's 3 ways to make an error, then you are in every time, for each one of those, there's a probability of 0.05 of making the error, and you do it 3 times, you're accumulating chances for making an error. Yeah, I'll leave it at that. So what you might do in a classical case like this is do a multiple testing adjustment of your p-values. My p-values are too small because I'm doing multiple ones of them. There are ways to adjust it so that at the end, even if you did take the minimum of the adjusted p-values, you can still say, I tested at a type 1 error rate of 0.05. In the context of omics data, we won't have 3 tests. We will have thousands of tests because we have thousands of genes, peptides, and other things. So this issue gets magnified in omics data. So I'm going to introduce you to an alternative way to assess confidence in a test. Instead of talking about type 1 error, we're going to talk about something called the false discovery rate. It turns out that it's more, it's kind of a more liberal way to gauge errors. It is designed to accommodate some errors as long as we keep the proportion of them sufficiently low. If we have thousands of features or thousands of genes and I want to know which genes are associated with Alzheimer's disease, we're going to get thousands of p-values, and I don't think there's only one gene in there that is important to this process. There's probably many, dozens, hundreds, maybe even. So I'm okay with making a few mistakes, getting a few false positives in my list to investigate further, as long as I get a good chunk of candidates that I can be confident in. So when we go to omics data and think about how to trade off possibilities of making mistakes, when we use a false discovery rate, we're saying, I'm okay with making some errors, I just want to keep them relatively low. Okay, a little bit of background, or just reminder. What is a hypothesis test? There's a consistent approach to doing a hypothesis test. Step one would be you write down a null and alternative hypothesis, which in our case might be the expression values, the numbers that we're using to quantify activity level of a gene. The mean of those differs between Alzheimer's and not Alzheimer's. The null would be that it does not differ. There's no difference. The alternative would be that it is different. Okay, so now consider one gene out of our thousands. And for that one gene, we have n observations. Maybe it's n over 2 for Alzheimer for treatment and n over 2 for placebo. So I can do a test. And the way we do that is we compute something called a test statistic. That's one number. I boil all those, my sample, all, let's say 30 people, 15 Alzheimer's, or 15 treatment, 15 control, boil all those down to one number. For example, that one number might be a two-sample t statistic. Sample mean minus sample mean divided by standard error. And the statistic is kind of our quantification of the magnitude of difference. How inconsistent is our collection of data relative to the null hypothesis? The test statistic is computed in such a way that it will be big in magnitude, absolute value, usually, if the alternative hypothesis is true. But then we need to know how big is big enough. Is a t-statistic of 1.82 big enough for me to be confident about rejecting the null? In order to answer that, you need the sampling distribution of the test statistic. Every time I go get a sample and compute a t-statistic, it's a realization of a random variable because the data were collected randomly. So if we were to imagine taking many samples, here's a sample, there's its t-statistic. Here's another sample, there's its t-statistic, and so on. If we did that thousands of times and drew a histogram of all the t-statistics, that would be the sampling distribution of the t-statistic. I specifically want to know the sampling distribution when the null hypothesis is true, because a p-value, again, is the probability that if the null is true, we'd see evidence like ours or even more extreme. So when we do a t-test, we compute our test statistic, 1.82 or something, based on my observed data. And then we need to know what is the distribution of a t-statistic when the null hypothesis is true. With a t-test, we know the answer analytically. It is the t-distribution. That's why you can look up in a table or something what a p-value is, because it's been figured out. But generally, what I need is this distribution. That's the sampling distribution under the null hypothesis. Okay. Once you have that, you can compute a p-value. How likely is it that you'd see something like what we saw or something even bigger, say, if this is the sampling distribution? It's just the area under the curve to the right of that point. So the key is to have this sampling distribution. That's the hard part. Then you compute a p-value, and you reject the null hypothesis if the p-value is small. All right. So there's two types of errors we can make when we do a test. I could, if the null hypothesis is true and I incorrectly reject it, I make a type 1 error. If the null is false, but I fail to reject it, I also made an error. We call that a type 2 error. When we do tests, we want to balance those as well as possible. You can't make both of them simultaneously small. If you make one really, really small, the other one will start to get really, really big. So here are some questions that come up in our context. Okay, so we're going to motivate this idea of false discovery rates in just a second. The first question we're going to ask is, suppose you do lots and lots of p-values, lots and lots of tests, what would you expect them to look like? There's actually an interesting shape that you expect for a bunch of p-values, and it can be used as kind of a diagnostic for data quality. We'll see that soon. Let's start here with this question. I'm going to come down to the section that corresponds to it, and let's make up some fake omics data. I'm going to make an omics data set M by N, where M is 1,000, so I have 1,000 genes, say. And I have 10 samples in each of 2 comparison groups. So we're about to create a 1,000 by 20 matrix of numbers. And to begin, we are going to make it so that there is no difference in the mean between the 2 comparison groups. So for my 1,000 features, the mean for the control group, same as the mean for the treatment group. I'm going to simulate under that. So making the 2 means, mu1 and mu2 for the 2 comparison groups, making them the same, I'm randomly generating what those means are. Doesn't matter, we're just doing a simulation. But the key is that I'm making them the same. So as we go feature by feature and do a test, the null hypothesis is true every time, because we're making it be true. Then I'm going to loop through feature by feature and just generate some random normal data. Standard deviation is going to be the same, just because we're playing pretend. So here are simulated data. (1,000 by 20). Here's the top. So might as well be the QC spike data. It's just numbers in a matrix form. Now let's do some testing. We are going to do a 2-sample t-test for each feature. So I have a loop over all 1,000 features. Do t.test. This is the control group. Ith row means the ith feature. This is the treatment group, ith row, but now the last n columns. And if you do t.test, you get several things back. If you do $p.value, you only get the p-value. I just want to look at the p-values. What happens when I do thousands of tests? So here will be 1,000 p-values, and here's a histogram of them. Looks like that. Of all the distributional names that you've ever heard, there's normal, there's gamma, Poisson, chi-square, and all that. What name would you use to describe that? If you remember. You know. What distribution does that look like? Looks like what we call the uniform distribution, which is just flat. This is a, there's random variation. If we redid this with another 1,000, they would shift a little bit, but qualitatively, this is just a flat distribution. Between 0 and 1, p-values follow this distribution when the null hypothesis is true. And you could show that that is actually theoretically the truth. The sampling distribution of p-values, the sampling distribution of p-values when the null hypothesis is true is that. What's the total area of that? If you were to like integrate that box, it's 1. The integral of any distribution has to be equal to 1, the area under the curve. That just defines where all the probability for these p-values is. What's the probability that you would see a p-value less than or equal to 0.5, do you think? If the total area is 1, then the area to the left of this would be 0.5, shading in everything to the left. What about 0.05? What's the probability that if the null hypothesis is true, you'd see a p-value less than 0.05 just by chance? If the area to the left of 0.5 is 0.5, what's the area of that rectangle? The width is 0.5 or 0.05, and the height is 1. So what's the probability that you'd see a p-value 0.05 or even smaller if the null hypothesis is true, just by chance? It's 0.05. What's another name for that? That's the type 1 error rate. That's why that's where we get this idea of type 1 error rates. If the null hypothesis is true, we expect p-values to just be uniform between 0 and 1. So I don't expect to see a small p-value, like less than 0.05, but it could happen. 5% of the time I expect to make a type 1 error. When I'm doing one test at a time, that's the story. All right. Now let's make things a little more interesting. File that picture away uniform, because we're going to use it a few times going forward, that uniform distribution of p-values when the null is true. What happens now when there are some different features, when there is some signal in the data? We are going to create, out of our 1,000 features, we're going to make the first 300 have differences, be differentially expressed. I'm going to randomly pick how different, but the bottom line is that we're going to take the data in the first, or in the treatment group, and add a difference. We're going to shift them so that now the null hypothesis is not true for these first 300, but it is for the last 700. So we're going to modify capital Y, and then let's go do the p-values again and look at what happened. Now it looks like that. And that, if you do testing, like differential expression analysis with omics data, you will see that picture a lot. If you do p-values for treatment versus control for my thousands of features, histogram p-values, you will see this picture a lot. What is this picture? We saw a second ago that if the null is true for all the features, that picture will be flat, because that's the distribution of p-values when the null is true. And for 700 of our features, that's the case. So behind the scenes, there is a flat distribution. The 700 p-values are contributing to a flat distribution. But what about the 300 features that we just put signal into? Where would their p-values tend to be, do you expect? Are they going to have big p-values or uniform p-values? They're going to have small p-values because they actually have differences. So their t-tests are going to be extreme. Their p-values, the areas under the curve, are going to be small. So this spike over here is due to the actual signal. We have a bunch of features. The majority of them, it looks like, don't differ between comparison groups, but there is a subset. And I'd like to go find the subset. So you can think about this picture, and we'll close with this that I'm about to draw. You can think of that picture as a mixture. We just saw something that looks like this. You can think of that as there is some proportion of the features, call it, and we'll use this notation again, that's why I'm going to call it something weird, pi 0. Some proportion of a flat distribution weighted with a distribution of actual signal p-values. It's like a mixture, taking those two and overlaying them. When we get the false discovery rates next time, we're going to basically start by looking at this picture. And we're going to say, somewhere in there are the features I would like to pull out and highlight. This represents how much kind of background noise there is. And we'll be able to systematically compare or assess that picture using some simple equations to essentially split into this and say, here's my interesting features, and now I can tell you something specific about how confident I am in them. I will, for example, be able to say, here, out of my 1,000 features, here's 220 of them that I find that I think are statistically significantly different. And just how confident am I in that? Well, the false discovery rate for that claim is 0.05. And what you get to interpret that as is, out of these 225 that you just handed me, I can say I expect no more than 5% of them to be false discoveries. You don't get to say that kind of thing if you work with p-values. I have more to say about that we'll get to next time. But that's some introduction to this multiple testing issue. We'll keep going next time. Let me just show you a couple of the things that we will also talk about. Some of this was requested by folks when I asked, what are you interested in? We will talk about technical versus biological replicates in this discussion, because in my omics data set, 20, or let's say 30 columns, 30 samples, is that 30 individual mice that we bred and subjected to a study, or is that 10 mice that we measured 3 times each? It matters, and I'll show you why. What happens if we have correlation between columns? Like I take measurements, genomic measurements, once every 6 months for every person in my study. There's correlation across the columns. What happens? And related, what happens if there's correlation across the rows? The genes in your study are correlated with each other. As this one goes up, these other ones tend to go up as well, or vice versa. I'll show you a couple of simulations that speak to impacts of those things. Okay, sorry for keeping you late. Questions before we go? All right, see you Friday.